My name is Jeremy Barr, and I'm currently a professor at Monash University in the School of Biological Sciences. And yeah, very happy to be presenting after Simon. I think that was a great talk, and I'm going to follow up and give a little bit of a different direction and perspective on how we can really tackle our understanding of these bacteriophages within environments such as the human gut. So jumping into my talk, I'm not going to spend long on this time given the audience that we're presenting today. We're all working in different microbiomes, but really the focus of my talk today is going to be on the most abundant, but also the least well understood entity in the gut microbiome, which is, of course, the viruses of the microbes, the bacteriophages, or the phages for short. And it's undoubted that sequencing has really revolutionized our understanding of the virome in these complex and diverse ecosystems. But for anyone who's worked in the phage field, you might have isolated and sequenced a phage genome or assembled a VMAG genome from metagenomic sequence stamps. You might relate to this point, and that would be you get your sequences back, you assemble, you annotate, and it comes back with over 80% of the genes being hypothetical and the taxonomy of that virion being novel and unclassified. And this makes it really challenging to understand the putative biology and function of these viruses in these ecosystems. So if I was to boil my point down into a single point, it would be that I really believe that we need additional work in wet lab biology to supplement the advances that we've seen in the sequencing and genomic space, particularly within the human gut, if we want to understand what these viruses are actually doing in these complex ecosystems. And so I really think, yeah, the gut is one of the next frontiers for many aspects of biology, phage biology included, but it's a really, really challenging environment to work in. We need entirely new techniques and approaches, and perhaps most limiting is that most of these gut microbes require culturing under strict anaerobic conditions. So this is a photo here of one of the anaerobic chambers that we have in our laboratories that allows us to grow and work with pure culture human gut bacterial isolates in this 37-degree anaerobic chamber. And I've also got to give another big shout out here to Associate Professor Sam Foster at the Hudson Medical Institute. This work is really a long-running collaboration between my lab and Sam's group. And we began working with Sam, and Sam's group runs the Australian Microbiome Culture Collection, or AusMIC. They actually have well over 4,000 strains now to date. Most of these in pure culture, all obtained from human gut microbiome samples, and many of these strains have whole genome sequence information available for them. So we began working with Sam. We felt that this was an incredible resource that we could utilize to understand the viruses that infect and live within these human gut bacterial isolates. So I want to spend like a minute or so just going over this very fundamental slide on phage biology, which shows the two major life cycles, which is, of course, the lytic cycle, where the phage infects, replicates within, and eventually lyses the bacterial host cell, leading to the release of the viruses, and the lysogenic cycle, where the phage infects, but instead of replicating and killing, it instead integrates its genome and resides and lays dormant inside its host bacterial chromosome. Now, most phage-based approaches are going to start all the way up on the top left, right? Isolating a novel phage from the environment and characterizing it. But what we did is we took a very different approach, and we started all the way down in the bottom right-hand corner of this slide. We began with these bacterial lysogens. Now, we call a bacterial a lysogen when it has an integrated phage genome inside its own chromosome. And because we have access to thousands of these human gut bacterial isolates from pure culture, we can use bioinformatic approaches, and we can predict and we can identify these integrated prophages inside these genomes. Now, we call a phage a prophage when it's in its integrative state, residing within the bacterial host chromosome. So we did this on a massive scale, doing these prediction softwares. But we also have access to the pure culture isolates. So we took this a step further. We went back into the wet lab, we grew these isolates, and we exposed them to a range of chemical compounds, stress conditions, metabolites, in an attempt to induce the prophage, induce them out of the lysogenic cycle back into the lytic cycle in a process known as induction, which is eventually going to lead to virion replication, host cell lysis, and the release of temperate phage virions. We call phages temperate, meaning that they can replicate by either the lytic or the lysogenic cycle, and they're temperate in nature. They haven't made up their mind which life cycle they're going to replicate within. So that's the overall approach that we took in this study. So what do we actually find? Well, I'm showing here an inverted phylogenetic tree of the 252 human gut bacterial isolates that we work with in the study. These were broadly representative of the diversity seen in the human gut. And just a reminder, we have all of these isolates in pure culture, and we have whole genome sequence information for all of them. So we began with the whole genome sequence information. We predicted high-quality integrated prophage sequences in all of these isolates. We found over 700 high-quality prophage predictions. 94% of the isolates harbored one or more prophage. And you can see in this inverted tree, many of these isolates harbored high numbers of integrated prophages. Particularly within Bacteroidota, we see several isolates harboring 5 or more high-quality predictions. These next gray bars here show whether or not that isolate was sequenced under one of our induction conditions. And then I'm showing you here the experimentally inducible prophages that we could reactivate from these isolates, finding 125 from these 252 gut bacterial isolates, with many isolates producing several integrative and active prophages. Several of these were very broad host range and abundant in the human gut as well. And the teal line showing prophages that were shared across species, and the black line showing prophages shared across genera. So some of these quite highly mobilized and prevalent in gut microbiomes. Now, in the interest of time, I'm just going to really tell you one short story from this paper, and that is looking at this discrepancy here. 713 high-quality prophage predictions, but just 125 of those, roughly 1 in every 7 of those, we could experimentally reactivate and reinduce in the lab. And that was a somewhat stark observation. It has been seen before, and we do know that the majority of prophages you can find bioinformatically in the gut don't appear to be reactivated. And so I want to introduce this concept of a cryptic prophage. A cryptic prophage is a prophage that's integrated, residing in its bacterial host genome, but it's lost the functional ability to re-enter into the lytic cycle and produce a functional infectious virion. It's effectively trapped in its bacterial host. This gives a lot of benefits to the bacteria. You get access to all of the prophage genomes without the threat of bacterial cell lysis from induction. And so we looked at our sample set. We had quite a number of samples from both active and these cryptic prophages. We pooled these and we performed a range of genetic analyses to look at what were some potential genetic cues that might explain this. So I'm showing you here a DNDS analysis, a ratio looking at the ratio of non-synonymous, predicted to be non-functional mutations, to synonymous mutations. These are across both our active and our cryptic prophage collections, and I'm breaking down these comparisons into these 7 to 8 functional phage gene categories. Now, what we found was that there was a significant increase in non-synonymous, potentially non-functional mutations in cryptic prophages in these 2 functional gene categories, one being the other, which are a bit hard to determine. A lot of these are unknown and hypothetical, but really interestingly, significant increase in non-synonymous mutations in genes associated with integration and excision. And this is important because these genes are the first genes that are required for the prophage to integrate and also to excise from the genome, and are really a key step from switching between these 2 life cycles. So from this, we propose an in silico analysis and mechanism that non-functional or non-synonymous mutations in integration and excision genes provide the first step to trap these prophages and make them become cryptic. Now, that's great in silico prediction. Can we take this further? So we went into the lab and we did some engineering approaches. We adapted a CRISPR-Cas engineering method, and we got this to work in a Bacteroides host. And we used this to delete one of these integrase and excision genes from one of our model prophages known as prophage POMA. Bit of a complex slide here. So on the x-axis, I'm showing you the genomic position in the bacterial host, this CCO1414 Bacteroides strain. And down below that, I'm showing you the genes and the ORFs of that integrated prophage POMA. Now, in the orange color, this is one of the experiments where we induced this isolate, and this particular phage responded to stevia and hydrogen peroxide. And you can see when we add those agents, you see this really large logarithmic increase in the number of viral genome copies along that prophage POMA genome, indicating that that prophage is being induced, replicating, and re-entering into the lytic cycle. Now, in the dashed black box there, we've actually used CRISPR-Cas to delete that DNA transposition protein or gene, which is responsible for excising the prophage out. When we knock that out in the blue strain, that's a clean deletion. You can see we absolutely abrogate the phage's ability to re-enter into the lytic cycle, and it's effectively trapped and become a cryptic prophage. So from this, we conclude that these non-functional or loss-of-function mutations in these integration and excision genes do provide a functional pathway and early step towards prophage domestication in the gut. Now, I'm almost out of time, so I'm going to finish on just some very broad conclusions. Again, this is just a very, very short snapshot into this study. And what I'm really excited about is this is the largest collection to date of experimentally validated gut prophages available. And I think these are incredibly valuable fundamental models that we can use to understand the diversity, the function, and the genetics of these understudied bacterial viruses. I also show you one example where we made a bioinformatic prediction of how cryptic prophages eventually become to exist in the gut, and we provide some functional wet lab in vitro data that we can knock out and engineer those prophages and confirm this putative mechanism. But I think most impactful and interesting is access to these collagen isolates allows us to study these phages in the wet lab. And in fact, many of these phages we found in our study had only before been detected in sequence space. So they'd never been found in wet lab. We've got some of the first in pure culture isolates of these phages. And I really believe that these prophages have large potential to modulate the human gut microbiome. And it's only through enabling these wet lab approaches that we're going to be able to realize that. So I'll wrap up there. If you're interested in this work, this was published in October last year in Nature, so you can go and read that further. Again, I just gave you a very small snapshot into that study, and I'll finish with some big thank yous. Big thank you to Sophia, who was the first author, Laura, who was the second author, the Foster Lab, all our collaborators and all our funders. Thank you all for listening, and I look forward to answering any questions you might have in the coming session. Cheers.